Hello, everyone, and welcome to our online lecture series for plant biology. Today, we start our journey in our exploration of studying plant diversity. And as you've hopefully experienced in your own life, plants are very, very diverse. They've got a lot of different life history strategies to live in the desert as cone plants or in aquatic environments as unicellular algae. And then those that you are more familiar with, some of our agricultural plants, like our flowering grains. The lab rat plants Arabidopsis, the mosses and liverworts that make up our non-vascular plants, the huge amount of diversity that we see in ferns, both our deciduous or leaf color changing trees, leaf shedding trees, and our evergreen conifers. And then some of our early aquatic flowering plants. Plant systematics and plant diversity is a very broad field that still produces lots of cool things beyond pretty pictures for society. We keep exploring plant diversity to better understand and identify new compounds for future medicines. Now, in discussing plant diversity, we're going to have to figure out a system of grouping species. And that's the entire goal of today's lecture. Understand some basics about how we as scientists put together plant histories in order to understand their evolutionary relationships. Because ultimately, all of these different plants, despite how different they look today, can trace themselves back to a shared common ancestor. And it is our task in this half of the class to put together that tree of life. In our effort to do so, we're going to first go over some vocabulary terms. The first is parsimony. And we will be using parsimony as our guiding principle in building our tree of life. Parsimony is the idea that you want to build the tree of life that requires the simplest explanation. It's also referred to as the principle of Occam's razor, wherein you use a razor to whittle away the nonsense and extravagant explanations to find just the most simple and therefore scientifically likely explanation for relationships between plants. Well, that's hopefully gives you an idea of what the term means, but it doesn't give you an idea of the applications of the term. In order to understand how we apply parsimony, we need to look at the difference between traits that are ancestral versus traits that are derived. And we'll do so by looking at this sketch of a phylogenetic tree. Now in this tree, we've got 5 different species, A, B, C, D, and E. And we have put forth this phylogenetic tree as a hypothesis for how they are related. This tree illustrates that A and B are most closely related to one another. They share an intersection of their branches together before they intersect with any of the branches of any of the other species. This means that they share a most recent common ancestor, MRCA. Now, when you look Further back in the history of this tree, we see that A and B share a common ancestor with C, and that D and E share a common ancestor with each other before any of them intersect with A, B, and C. Without looking at the back half of the tree, so if we were theoretically going to ignore that section of the tree, we have two different groups. But then, with the incorporation of that back, we can see that the groups A, B, and C can trace their lineage back to a common ancestor with D and E way back here. This allows us to establish which species are more closely related to one another. Species that share a most recent common ancestor are considered sister taxa, meaning that they're right next to each other on the family tree. You can additionally say that C is sister to the group A and B. It just means that the next branch will intersect with a shared most recent common ancestor. Now, how do we today know that A and B are sister to one another? Do we put these ideas forth without any evidence? Of course not. We build these relationships based on matrices of traits that are ancestral versus derived. An ancestral trait is some feature that we'd expect to find early on in the tree. When a trait is ancestral to the tree, like let's say having purple flowers, we therefore assume that this purple flowered trait is inherited by all of the descendants of this tree. At some point in their life history, species A, B, C, D, and E would have had an ancestral state of purple flowers. Now, we know that evolution occurs. We know that evolution drives changes. And perhaps purple was beneficial and stayed purple in the lineages of A, B, and C. But D and E share a history in a different environment where the pollinators may not have seen purple and therefore may have shifted away from their ancestral state to a new orange color. Now, this new version, changed from the ancestral, is our derived state. To illustrate the principle of parsimony, we can see how many tick marks or how much evolutionary currency we need to expend in order to have the flower color of D and E shift to orange. What I mean by this is that we want to be illustrating our traits not by those arrows, but rather by tick marks. Ancestral being purple, and putting a single tick mark on the tree costs one evolutionary currency, one mutation, right? Because ultimately these are all mutants of different things. And so in order to have a purple color, we will spend one tick mark to state that all of the species A, B, C, D, and E have an ancestral state of purple flower color at the base of the tree. Now, if we want D and E to inherit a orange flower color, we could have D inherit and evolve orange, and E inherit and evolve orange separately. But that costs us 2 tick marks. Instead, we could be more parsimonious with our explanation and say that the common ancestor of D and E evolved orange flowers, and that D and E are inheritors of this different, derived character state. In this way, we have now constructed a more parsimonious, a simpler explanation. Rather than having 2 separate events for D and E to change colors independently, we now can say that our best hypothesis is instead that their common ancestor evolved orange flowers and both of them inherit this novel state. Now, of course, if we have better evidence to suggest that these evolutionary transitions were actually separate, then that better evidence would be incorporated and our hypothesis would change. And so you will notice that our phylogenetic tree of life does shift throughout time. It has changed from when I first took biology to now when I'm teaching biology. The tree of life changes a lot as we continue to accumulate more and more evidence. So hopefully that makes sense of our terms of ancestral versus derived and parsimony. In order to illustrate our next terms, homology and homoplasy, we will again turn, we will again turn to our tree of life. Howdy settle. A trait that is homologous is one that is derived from shared ancestry, meaning that purple, or even the ability to have flowers, is a trait that's homologous to our tree here. Now, a trait that is homoplasious is something that's evolved by convergent evolution. Convergent evolution and homoplasy are 2 things that will complicate our ability to build better trees, or build more accurate trees of life. Convergent evolution occurs when 2 not related species end up looking similar. This means, in terms of our toy example here, that you may see orange flowers on something like species B. If species B has orange flowers, you may construct a tree whereby the purple A and C are grouped together, and then they share a common ancestor with D and E, and that D and E are closer related to B than they would be to A and C. I'll redraw this line, sorry about that. This would allow us to group our orange flowers with our apart from our purple flowers. This may have occurred because, let's say that B uses a different pollinator than A and C, who prefers orange flowers rather than purple flowers. This may occur by random chance. We can do a lot of experiments to try to figure out why this might happen, but this instance of convergent evolution does complicate our ability to construct a phylogenetic tree that is accurate to the underlying truth. Now, we need to remember that this flower trait is but one trait. Even though you can group these species based on flower color and look like this, once you start incorporating other features like leaf shape or seed size, or any number of different things, they would then contribute more signal to your tree, allowing you to better construct a tree of life that recapitulates what actually happened in the past. Because all these trees of life are trying to capture things that happened before humans were a species on planet Earth. And while we can't go back in time, we can look at the evidence that time has left us to try to construct a better tree of life. So cases of convergent evolution are where we see similar phenotypes without shared ancestry. And therefore, convergent evolution requires us to spend extra currency, extra tick marks, in order to make the explanation. So going back to our tree here, we would instead have to spend one evolutionary mutation here on the shared ancestor of D&E to give D&E orange flowers, and one tick mark here on the shared ancestor of just B in order to differentiate B from A, in order to switch B from those purple flowers in order to those orange flowers. And so homologous traits would be ones that are shared by ancestry, and homoplagious traits, or a homoplasy, is a trait that's shared by current appearance. And we will spend a lot of time talking about traits that are similar in different plant species by convergence, and comparing those traits to traits that are shared across plant species because they all inherited the same ancestral state. Now, one thing that I will note in these drawings of our phylogenetic trees is that for the purposes of this class, the length of the branches is meaningless. Now, that doesn't mean that in all phylogenetic trees branch length is meaningless, but rather than branch length here in this class is not going to correspond to evolutionary distance. There are trees drawn by computers that have huge data sets for which branch length does have meaning. And what I'm trying to illustrate here is that you can't say that A and B are more closely related to each other than D and E, despite the fact that A and B seem to have less distance between them. Know that that is just an artifact of me drawing them as a human freehand. If we had boatloads of data, whole genome sequencing, phenotypic matrices, you may be able to better estimate the relatedness of A and B to one another versus the relatedness of D and E. In fact, that was one of the things that I did very commonly in my PhD. But here, I don't want you to draw interpretations based on my drawing abilities. So I want to point that out before we get into our major trees of life. A couple other terms are illustrated here on this cartoonish tree of life without any endpoints. A polytomy occurs when we cannot determine which species are more closely related to one another. We may know that the species here, species A, B, C, and D, all share a common ancestor, but we don't know whether A and B are more closely related to one another, or that A is more closely related to C, or maybe B is more closely related to D. We do not know how the structure, the hierarchy exists in this group, but we can illustrate that they all share a common ancestor by collapsing their branches into a polytomy by connecting them at one simultaneous point. So imagine yourself tracing this back. I'm going to run my finger down A's line and see which other species it intersects with. Well, it intersects with B, C, and D all at the same time. Therefore, we have this polytomy. And we will see polytomies even to this day in our trees of life, where we are uncertain as to how this diversity relates to one another, but we know that they all do share a common history because of an overwhelming amount of ancestral traits. This is particularly true in our ferns, where things that don't look quite like ferns will still group with our ferns. We also will refer to the traits that are close to the root, close to the base of the tree, as our basal taxons, meaning a species that can be fairly archaic in how it looks. The reason why these basal taxons look evolutionarily old is frequently because they split off and retained the ancestral traits from the root, but did not change the exact same ones that were occurring on the other branch. These branch points allow species to coexist on planet Earth and evolve independent from one another. They allow these species, or at least us as scientists, to illustrate how these connections occur in the past and show that some of these ancestral traits can sometimes be, quote unquote, frozen in time in our basal taxa, like some of our mosses and some of our liverworts. These are plants that have no veins. Before plants had veins, they had to not have veins, as veins are a complicated, derived state. And so in looking at some of these basal taxa, like liverworts and mosses, we as scientists can get a better idea of what the history of plants looked like. What did they look like when they first came to land? There's another bit of terminology that I want to cover, because not all of the species that we study today are alive today. Some of them are extinct. And we can use these fossils, these once living, once abundant species, in order to help guide our relationships in the branch points of our living plants today. The plants that are living today, and animals, anything that's still alive, is considered extant. Extinct, no longer here, but we may have evidence of it in a fossil, versus extant, alive, today. And we as plant biologists will very frequently use the fossil record to help us get a better idea of what species looked like in some of their ancestral states. It's also important to understand that species that are our basal taxa continue to evolve today. And so, even though things like mosses and liverworts don't have vascular structure like some of our early land plants, they have a lot of features that have evolved since that branch point with our early land plants. Their evolution has not frozen in time. They will not resemble exactly our extinct plants. They may have a lot of clues in their morphology as to what were some of those shared ancestral characteristics. So this is some of the terminology surrounding how we build our phylogenetic trees. We also today want to discuss some of our species concepts, because it can be really tricky to try to identify what makes two different plants members of the same versus members of different species. This is particularly true since plants are so willing and ready to hybridize. They are very fluid in their ability to make progeny with very distantly related taxa. For purposes of this class, I'll introduce you to three different species concepts. The first is the biological species concept. In the biological species concept, we consider two different species when they can't make viable offspring together. If they can't make babies that can then go on to make babies, the biological species concept separates them into two different taxa, into two different groups. Pretty straightforward, except for in plants that we can have two different species that will cross if forced, but may not do so in the wild. So our biological species concept can get muddled. The next is the phylogenetic species concept. By the phylogenetic species concept, you are trying to group everything that has a shared ancestor into a single clade. So if we're looking at different populations, as long as all of them can share direct ancestry and still can produce offspring, even if they don't do so today, we can consider those species the same by the phylogenetic species concept. Bless you, Odie. I don't know if y'all can hear the dog sneezing in the background. He's being quite rambunctious today. Our third and final species concept that we'll cover in plant biology and botany is the morphological species concept. And in the morphological species concept, species need to look distinct. Their flower shape needs to be different and genetically imparted. Their leaf shape needs to look different and be derived from differences genetically. Maybe they can make babies, maybe they can't. Maybe they do group into one shared common ancestor, maybe they don't. But by the morphological species concept, we can differentiate species based on whether they look different. And so no one concept is perfect. No one concept is universally accepted. Different environments, different situations, different species even, will utilize the biological, phylogenetic, and morphological species concept in order to differentiate groups. Now, one of the ways in which groups will differentiate from one another is by adapting to different ecologies, adapting to different environments. When we have members of a species that are adapted to a specific environment, we'll start a new subheading here for adaptations. We consider a group of a species adapted to a specific environment an ecotype, because they are the group or subset of a species that is adapted to a specific ecology. And in this class, we will actually be taking a look at one of the first scientific studies that tried to classify ecotypes of a couple of different species in California. You'll notice the Sierra Nevada mountain range and their sampling sites, everything from samples in Stanford, before it was, it was established as a good school, but before it was the juggernaut that it is today, to the peaks of the Sierra Nevadas, where a lot of different ecotypes are adapted based on differences in altitude. Ecotypes, and our discussion of ecotypes in that paper later, will focus around the fact that these plant subsets can be adapted genetically, meaning that there's no other option for them to be adapted to that environment because their genetics are fixed, they're obligated to live there, or whether they can flex and still thrive in other environments. When a plant has the ability to display different phenotypes based on different environments, it is said to have genetic plasticity. Genetic plasticity means that the plant species has the genes in order to adapt to maybe a wet environment and a dry environment. It maintains that ability to flex the way it looks, the way it grows, depending on the environment that it's in. This is always expensive metabolically. Just like you, when you're trying to do two different tasks, multitask, it is more difficult to accomplish two separate things at the same time than accomplishing one of those things. Takes more energy, takes more focus, takes more attention. Plant adaptation to different environments operates on a lot of the same principles. Traits that are plastic are oftentimes not correlated with ecotypes. Because that plant isn't particularly adapted to one ecology, it can flex between the two. Instead, traits that are genetically assimilated are cases where the plasticity is lost. And the environmental response or the growth response is fixed, meaning that it's stuck, it's glued, it's one type, not the other. Can't be flexed back and forth. And so when we're talking about a lot of these features on a population level, and we're referring back to our species concepts, we will see that some ecotypes that we classify as the same species, because they've assimilated into different environments, that they may not normally make babies together, they may not normally mate, but we can force them to do so in the lab, and they may be somewhere on their way toward true speciation, but not there yet. This serves to remind us all that evolution is an ongoing concept. It is not frozen in time within our lifetimes. Yes, it takes a long time, or can take a long time to occur, but it is still ongoing. Even we as humans are still evolving. And this certainly is true for our plants today. Now, when species have fully separated or fully speciated, two of the very common ways in which this can be accomplished are either allopatric speciation, this being the more common method, or sympatric speciation, this being the more rare method, particularly in plants, because up to date, there's only been one scientifically proven example of sympatric speciation within plants. And you guessed it, it is between these palm species on Lord Howe Island, and we will be reading the article published about this case in this class. Allopatric speciation occurs when species are in different environments, or using the roots of the words, allo, meaning different, and patries, or fatherlands. They are in different fatherlands. And when they exist in different fatherlands, they're adapting to different climates. Perhaps they originated from one place, but a river ran through that place, and now one side is flooding constantly and the other doesn't get any water anymore. So now you've got wet versus dry. Or perhaps one is even submerged, or one has a different soil type, one has had a volcanic explosion. There are lots of situations that cause environments to change from what they once were. And species that exist for long periods of time in different environments are likely to speciate via allopatric speciation. In contrast, sympatric speciation is where species will differentiate by living in the same place for long enough. They speciate in the same environment or same fatherland. This is particularly rare in plants, less rare in animals, which can speciate via behavior, things like mate selection or something like that. But there are still, or at least one noted case of sympatric speciation in plants, where these two palms have existed on this island for a long period of time, and we will be talking about what evolutionary forces that would have driven the Kentia palm and the Belmore Island palm to be distinct species. We will additionally be talking about cases of adaptive radiation. Adaptive radiation can occur in plants when one original colonizer will arrive to an environment where there are not a lot of competing species, where there are a lot of ecological niches that are open. And from that one colonizer, they can differentiate into lots and lots of different environments. This can occur over relatively short timescales. One of the best studied cases of adaptive radiation is the Hawaiian silversword. Hawaiian silverswords are a very species-rich group. There are a total of 28 different species that nowadays range from being trees to shrubs to vines to small little plush plants that you might consider a pillow to other different varieties. This is one of the most characteristic species of silverswords, named silverswords because their leaves are silvery in color and quite jagged. And based on genetic work and phenotypic work, we know that these Hawaiian silver swords arrived in Hawaii about 6 million years ago. As a single species, but then diversified into lots of different environments on the Hawaiian Islands. They allopatrically speciated by fitting themselves into lots and lots of different niches, such as the volcanic rock on one side of the island, maybe a well-developed soil on another, maybe a shady versus a drier spot on the island. These Hawaiian silver swords are very, very, very prolific, having diversified into 28 different species over the course of 6 million years, from what we can see is a single common ancestor. It's likely that this common ancestor came from Western North America, as its closest relative today are California tarweeds. So this case of adaptive radiation is where you'll have a single species that differentiates, frequently allopatrically, by fitting into lots of different niches in this new environment. Perhaps the best known case of adaptive radiation is, of course, Charles Darwin's finches of the Galapagos Islands, where one bird species flew to a lot of different islands and adapted to eat the seeds of each island, and therefore had changes in their morphology based on ideal beak shape, beak size, and eventually corresponding changes in overall bird size, mating call, and the rest started to occur. Now, another major driver, let's go ahead and move this down here since adaptive radiation applies to that, of speciation in plants, whether it's allopatric or sympatric, are whole genome duplications. We'll talk more about why whole genome duplications are so common and so important to plant speciation later in the class, but I do want to familiarize y'all with this basic concept that plants will very frequently have a polyploidization event. This occurs when a plant, rather than dividing properly via meiosis and going from something that's a diploid to making an egg and a sperm that are each haploid, it will instead retain diploids and they fuse with other diploids and create plants that are now tetraploid or octoploid or hexaploid. Plants have all kinds of different examples of this. These whole genome duplications are very important in plant history because they allow for extra copies of genes, for mutations to accumulate, for basically evolutionary fodder, regions of the genome that you can play with to try to optimize the different environments. And this plant tree of life shows known whole genome duplication events as red dots on the tree. And we can see that within your monocots, your grass relatives, these are all different grasses like corn and rice and wheat. We see that they all inherit lots of different whole genome duplication events. We see that the same is true for a lot of our dicots, things like tomatoes and potatoes. There's a whole genome duplication event that's specific to the potato group that we don't see in the tomato group. To some of the species that I studied during my master's, poppy plants. Whole genome duplication events and polyploidizations are rampant across the plant tree of life. Compare that to the animal tree of life. Not so many. In fact, only 3 well documented cases, and not really any occurring within the reptiles or mammals. Animals can't tolerate whole genome duplication events very well. Plants can. And there are two different types of whole genome duplication events. Auto polyploids, meaning whole genome duplication events that are self-fusions, meaning one species will double its genome, and that'll be that. It will, from there, potentially form a whole new species, as we see kind of like this. In allo polyploid events, you can get the diploid gametes from two very different species to fuse, and they will form a brand new shape, a brand new species that is still a polyploid, a fusion of two diploids, but it's also a hybrid from different species. So there we have our differences between our auto, meaning self within a single species, and allo polyploids of other species. You will notice that we, as scientists, do reuse our word roots a lot. Allopatric speciation, allopolyploidization. Hopefully it's helpful to keep track of all this. And that's kind of where we'll leave the lecture for today. We've got a good understanding of our different terms, different drivers of evolution, whether they be whole genome duplications or adaptive radiations, or allopatric or sympatric speciation, the underlying population effects, like ecotypes that are assimilated to an environment, or a broadly adapted species that is plastic based on the environment, how we define our species, and most importantly, how we draw our phylogenetic trees. I cannot emphasize to you enough how important it is that you understand how we make these connections, how we trace our trees based on most recent common ancestors, how we lay out traits based on being ancestral derived, and how we ultimately try to construct the most parsimonious explanation, the most parsimonious, succinct way of looking at the history of evolution of plants on planet Earth. So I'll end today's lecture by showing you where we're going. I think it's kind of useful to understand some roadmaps for our plant trees of life. A couple of different depictions. This from Wikipedia, looking at plants as a fusion of some sort of protozoan, a protist, a eukaryote, and cyanobacteria, these being our chloroplasts, that gave rise to a lineage that shares ancestry with red algae, that we have modern day living green algae, our charophytes being our closest relatives, our early land plants, the liverworts, mosses, and hornworts, collectively known as bryophytes. We'll have a full lecture on those. Our next lecture will be focusing on the charophytes and chlorophytes, and then looking at the plants that have vascular tissue, our ferns and their relatives, and then our two different lineages of seed plants, the cone-bearing seed plants and the flower-bearing seed plants. If we want to take a look at how to draw this tree with some traits, some tick marks, we can look right below it. A shared common algal ancestor. The differences between our charophytes and our liverworts can be a trait like embryo protection. Liverworts differ from mosses and hornworts by having shoot apical meristem growth. That's right, apical growth is very ancestral to all land plants. Makes sense why it's one of the things that we covered earlier in this class, right? And then, of course, vascular tissue being super important to plants growing tall. Leaves with lots of veins, megaphylls. And then the evolution of seeds. And then all of the different traits that we see in terms of flowering plant development. Now, this is a miniature version of what you will be producing by the end of this section of the class. A tree of life for all plants with traits as tick marks, particularly highlighting some of our convergent evolutionary traits. And so you might ask, how many branches? Well, not this many branches, even though you'll notice that this tree doesn't have a single branch or compiles all flowering plants into a single branch, despite the fact that right now on planet Earth, there are more flowering plants than anything else. But it does include a lot of extinct species. Yellow's everything that's extinct. Teal is everything that's extant. And this was from that class that I was mentioning to you, or we'll be talking about a fair bit, looking at the possible relationships across the entire plant kingdom. Thank you, Judy Jernstedt, for this. I'm sure you'll get some stories out of me about her in this class. Our tree of life will end up looking like this, with some relationships between our flowering plants, our cone-bearing plants, our ferns, our lycophytes, probably something that you haven't seen as of yet, unless you're a big fan of Star Wars and the planet Dagobah, and then where we'll be beginning with some of our early algal ancestors. So see y'all next time.